Um, I'm Christy Landis. I'm at Rice University, and the co-hosts for this webinar are Rodrigo Noriega at Utah and Carlos Baiz at University of Texas, Austin, and Zhonghui at Boston University. And we are very, very happy to be hosting another physical chemistry webinar with Professor Miriam Freedman from Penn State University, who is also, we're very happy to say, the awardee of this year's ACS Physical Chemistry Division Experimental Award for more junior faculty. The Phys division makes 4 awards essentially by the division, a theory award and an experimental award, and 1 for more senior faculty and 1 for more junior faculty. And so we're very honored that Miriam agreed to give a award talk here. In addition, right, you're still going to give your award talk at the ACS meeting, correct? Yeah, perfect. These webinars are sponsored by Phys Tech, which is a company that manufactures lasers and spectroscopy systems. And so we thank them for their sponsorship. And if you want to see the future talks, such as on June 25th, the talk from Greg Hartland at Notre Dame, you can go to the newly designed physical chemistry website at phys-acs.org to learn more about that or other meetings or anything like that. And so Rodrigo, Carlos, QC, did I miss anything? Okay, so now I'm going to stop sharing and introduce Professor Freedman. And Miriam, I didn't actually ask you where you're from originally. I know your educational background, but where are you from?
I'm from Baltimore, Maryland.
Oh, interesting. So a neighbor I'm from Virginia, so you're sort of your neighbor. Miriam has a really interesting academic background. It's not completely standard, I think, for a chemist or physical chemist. So she went to Swarthmore for undergrad, but then she did grad school in math and applied math, right? At Minnesota, and got a master's thesis there. And I don't remember the person you got your master's thesis with, because of course it's a little bit outside of our field, but then you transferred to the University of Chicago, right, to do your PhD. And that's with the name that most of the physical chemistry people heard of. She worked with Steve Seibner for her PhD thesis at the University of Chicago. And then she moved to the University of Colorado in Boulder to another physical chemist that we're all familiar with, who's Margaret Tolbert, to do her postdoctoral work after that. And then she moved to Penn State for her junior faculty career, has been promoted to associate professor, and so has a lot of really nice work that she's going to tell us about today. And what will happen is we will be asking for your questions in the chat feature, and you can type in your questions, and afterwards we will help curate your questions. But for now, I'll just turn it over to Professor Freedman. You go ahead and share your screen and start your talk.
Okay, so thank you, Christy, for that great introduction. I'm really excited to be able to give this talk today. And so what I want to do first is I want to give just a little bit of background about what my group does in general. And then I'll tell you about the project that I'm going to talk about today. So in general, my group looks at phase transitions of aerosol particles. Phase transitions and phase diagrams are something that's been interesting to me basically forever. It's one of the reasons that I went into chemistry in the first place, and into physical chemistry in particular. And so I'm going to talk today mostly about liquid-liquid phase separation, but my group also does a lot of work on ice nucleation as well. And so I will give you a little bit of background of what we do in ice nucleation. And so I want to start just by talking about the background of the slide that you're looking at now. So this is a picture of the Nittany Valley, where Penn State is located. Penn State is off to the right of the picture. And there's actually a lot of atmospheric chemistry in this picture, which I'll describe in a moment. This picture was taken by a former RU student in meteorology who liked to go storm watching with her fellow students. So this is a non-stormy day, viewing the valley from one of our surrounding mountains. So before I talk about the chemistry going on in this picture, I'm going to take a step back and tell you about atmospheric chemistry in general. So basically, at the surface of the Earth, there are a lot of processes that are generating molecules that are in the gas phase that enter the atmosphere and particles that enter the atmosphere. So for example, we can have forest fires, we can have emissions from vehicles, we can have agricultural emissions. There are winds that move over arid soils that loft mineral dust into the atmosphere. There are wave-breaking events on the ocean that result in sea spray aerosol being ejected into the atmosphere. And there's vegetation that emits a variety of different organic species. And so basically we have all these gas phase and particle phase species. And because our atmosphere contains oxygen, they get oxidized, they react, they interact with water. And so we get a huge complexity of thousands of different compounds. And looking at what those compounds are, what their transformations are in the environment, and what the physical properties of both the molecules and the particles are, is the field of atmospheric chemistry. So, for example, in the picture that I showed you to start, we have all this vegetation in this area, and that's emitting isoprene and terpenes. These get oxidized. They form organic compounds of various volatilities. And then some of those compounds go into clouds and get processed by clouds. Some of them condense on aerosols that are already in the atmosphere, and some of them nucleate on salt particles that are always in our atmosphere. And so we get the formation of secondary organic aerosol. And what I'm going to talk about today are the phases of that, what I'm going to call organic aerosol, but keep in mind that organic aerosol contains organic compounds, salt, and water. And before I tell you all this research, I want to thank the people who have done this work. So I'm listing here my current and former graduate students and postdocs, and I have a very old pre-COVID picture shown as well. And anyone who's either bolded or highlighted in a color, those are the students and postdocs who have worked on my liquid-liquid phase separation project or projects that are very similar in nature. And so when I show you the background of this project, that's going to be the work of Mohammed Altaf and Dan Vecti. And then I'm going to highlight the research of Teresa Kuczynski, who was a PhD student in my group who graduated last year and is currently a postdoc at NOAA. And she did her projects with help from Joseph Dawson and Emily Ott. The work that I show in the background was funded by Penn State and an NSF Career Award. Some related projects that we do currently are funded by NSF, and all of our physical chemistry of liquid-liquid phase separation work is funded by the DOE. Okay, so why study aerosol particles? Well, the World Health Organization considers air pollution to be responsible for 1 out of every 8 deaths per year globally, making it the most lethal environmental hazard. And particulates are an important component of air pollution. So air pollution affects visibility, for example, and that's shown on the left-hand part of this slide. These are pictures showing images during the COVID shutdown. So the top picture is showing an image of the Himalayas, which was visible for the first time in 30 years from 200 kilometers away due to shutdowns that resulted from COVID. And then the bottom picture is showing Long Beach, California. The left-hand picture is during a very polluted day in 2017. The right-hand picture is showing a picture during the shutdown. So you can really see from these pictures how air pollution affects visibility. It also affects human health. So for example, there was a famous study done in 1993 that's been followed up by many additional studies called the Harvard 6 City Study, published in the New England Journal of Medicine. And that looked at 6 different cities in the US and different pollutant concentrations. And what they did is that they looked at mortality rate which is shown on this axis, on the y-axis. And on the x-axis, they plotted the pollutants. And they only saw correlations between mortality rate and the pollutant level for fine particles and sulfate particles, which are, there's an overlap in those two categories. And in general, particulates less than 2.5 microns in diameter are associated with cardiopulmonary disease. And so why is that? So that has to do with where particles of different sizes go in the respiratory tract. And so when we have particles that are larger than a micron in size or smaller than like 10 nanometers, they can get trapped in our upper respiratory system. So for example, if you're standing next to someone who has the flu and they sneeze and you ingest one of those droplets or inhale one of those droplets, it can get stuck in your upper respiratory tract and you can get disease transmission through that route, as I'll talk about in a moment. If you have particles that are less than about 100 nanometers, they can deposit in the tracheal region. And if you have particles that are less than a micron or less than 2.5 microns, they can get into the pulmonary region. And so that's why particles less than 2.5 microns in diameter are associated with cardiopulmonary disease, because they cause inflammation of the lung. Chronic inflammation causes disease. So we can turn this on its head a little bit. So I've been talking about particulates that come from mineral dust and sea spray and vehicle emissions and things like that. But we can also talk about our own emissions. So anytime we do anything with our mouth, we're emitting aerosol particles and droplets. And so these are composed of respiratory fluid. Historically, the infectious disease community has differentiated between droplets, which are large in size, and aerosol, which are small in size, but they're really a continuum of droplets, a continuum of sizes of particles being emitted. So things that are large, like droplets, are going to gravitationally settle within 1 or more meters. That's why we have social distancing guidelines. Aerosol can remain suspended in airflow for long durations and travel long distances. And settled viruses can remain on surfaces, and we can, if we touch an infected surface and then touch our mucous membranes, we can transmit disease that way. So, you know, everyone's interested in COVID. So in terms of COVID-19, we know that most people who infect others through these larger droplets, that they're infecting 2 to 3 other people. But there are super spreading events where there are large numbers of people being infected. And these events are thought to be caused by aerosol particles, by transmission of aerosol particles. And if we can prevent transmission through aerosol particles, either through mass or ventilation or vaccines, then we can stop a lot of the spread of COVID-19. Okay, so returning from health to other reasons that we care about aerosol particles. The other main reason that we care about aerosol particles is because of their effect on climate. So our climate is determined by energy balances, how much energy comes to us from the sun versus how much leaves from the earth. And so you can see from this picture, when the radiation from the sun reaches us, about 30% is reflected off clouds in the atmosphere and the surface of the earth. And the rest is absorbed by the earth. And so the earth has a temperature. It also emits radiation. And some of that radiation goes back out into space. Some of it's captured by greenhouse gases. And it's good that we have a greenhouse atmosphere, because otherwise the temperature of the earth would be too cold to support life. But of course, the problem that we have now is that we're putting more and more greenhouse gases into the environment, which is causing the temperature of the earth to continue to rise. So what you can see from this schematic is that the atmosphere is a really crucial part of the climate system. And so it turns out that you can take every component of the atmosphere and quantify how much of a warming or cooling potential it causes. And that quantification is called radiative forcing. So this is a schematic from the last Intergovernmental Panel on Climate Change report. And it's showing many different components of the atmosphere and how much of a warming or cooling they cause. And all the values are referenced back to 1750. because we're trying to understand the effect of humans on the Earth system. So we're referencing back to pre-industrial times. So the first item on this chart is CO2. CO2 has a positive radiative forcing, meaning it causes global mean warming, which is what we would expect because it's a greenhouse gas. And we've also been putting more and more CO2 into our environment since pre-industrial times. You can also see that the uncertainty, the error bar is the uncertainty. You can see that that is relatively small. And that's because CO2 is one compound. We can put it in an IR spectrometer and measure its absorption cross-section, and that more or less explains what it's doing in our atmosphere. As you go down the schematic, though, there are two effects of aerosol particles that I've circled. So the first of these is how aerosols directly interact with light. And the second is how aerosols interact with clouds, which then interact with light. And these have a net cooling effect. They have a negative radiative forcing, but the magnitude of their effect isn't very well understood. And that's because directly the schematic, that's because global climate models of different types don't agree on these effects. And that indicates that aerosol chemistry and physics isn't well represented. And that makes sense because we're dealing with myriad different compounds. There are a lot of complexity of interactions. And when we have a global climate model, we can't put in all the chemistry and physics. Things have to be simplified in order to get the model to run on a reasonable timescale. And so our role as chemists in trying to understand aerosol effects on climate is to uncover all that complexity and then figure out where can we simplify versus where do we need that complexity to understand the situation. So I will talk about liquid-liquid phase separation, but I do want to tell you about what the other half of my group does. So I'm really interested in general, both in phase transitions and in cloud formation. And so when we have clouds forming in the atmosphere, we never have liquid water coming out of the gas phase and forming a cloud droplet. We always have nucleation on an aerosol particle. So we can have salt and organic compounds that take up water and form cloud droplets. And I'll talk to you a little bit about that later in the talk. Or we can have insoluble particles where ice is nucleated using these insoluble particles, and we form ice particles. And clouds are either made out of liquid water or ice or a combination of the two. When we look more carefully at ice nucleation, two different processes can occur. We can have that aqueous cloud droplet freeze, but that won't actually occur until -38 degrees Celsius. Even though the freezing point of water is 0 degrees Celsius, you need a defect in order to get water to freeze around that defect at 0 degrees Celsius. And so if you don't have a defect, like in an aerosol particle, you can't get the nucleation to occur until you can overcome the activation barrier to form that crystal. And that won't occur until very low temperatures. So if you're above this temperature, you always have to have an insoluble particle acting as your defect site to get ice nucleation to occur. And it can occur through a variety of different mechanisms. And so what we're looking at in my lab is how characteristics of surfaces promote ice nucleation. So there are many different aspects of surfaces that could be important in this process. So we can have crystallinity and lattice spacing. Defects or functional groups be important for the formation of ice. And what we want to answer is, we know that all of these are important, but we want to be able to predict the ice nucleation activity for a given surface. And so what we do is we use synthetic materials where we can tune these different properties and then look at the ice nucleation process and infer what was important about the surface for that process to occur. So I'm going to return to this picture now of looking at these phases of organic aerosol and give you the background that we'll need for as we delve further into this area. So first, I need to tell you about what traditional phase transitions have looked like for organic aerosol. So when you have a salt particle or a particle that's composed of salt and organic compounds, as long as you don't have too much organic or too viscous and organic, this phase behavior will hold true. So when you're at very low relative humidities, so we're thinking about phases in terms of the amount of water present rather than pressure and temperature. because that's what's relevant in the atmosphere, in the lower parts of the atmosphere. So at low relative humidity, the particle is crystalline or solid. And then as you raise the relative humidity, eventually the Gibbs free energy of having the particle and the water vapor is the same as having the Gibbs free energy of the aqueous particle. And so you get water to be taken up by that particle. And as you continue to raise the relative humidity, the particle will continue to grow. And if you start decreasing the relative humidity, the particle will enter a metastable state until it can overcome the activation barrier required to form a crystal. And then at that point, it will crystallize. It will go through efflorescence. Right.
So
and on the right,
Miriam, I guess this is so interesting because it's a different field from that normally we don't interrupt, but I'm going to interrupt just to ask a question here.
okay.
When we think of phase transitions and other parts of chemistry, it's really important to, the surfaces are wildly different depending on the other variable there. So you talked about the temperature in the slide before. Does this smooth phase surface continue regardless of the temperatures ranges that happen in the upper atmosphere, or does it strongly depend on that as well?
So that's a great question. And so most of the time when people do experiments looking at efflorescence, deliquescence, they're looking at ambient temperatures. But when people look at ice nucleation work, then they are incorporating temperature into these studies, and they can still be looking sometimes at deliquescence and efflorescence during those studies. I'm not sure that I've seen plots comparing temperature for these for like a plot like on the left-hand side. But so I'm not sure if they look exactly the same or what the differences are, but I know that you'll still have like this hysteresis loop between deliquescence and efflorescence. But that's an interesting question to look more into. Yeah, so on the right-hand side, I'm showing some experimental data from my lab, just looking at a particle that's effloresced and a particle that's deliquesced. So this one looks, the effloresced particle looks solid. The deliquesced particle has taken up water. Okay. In 2001, it was proposed, and then in 2006, it was actually shown that we actually have to consider two more phase transitions when we're dealing with solutions of salt and organics. That instead of just having efflorescence and deliquescence, we need to consider that organics can have limited solubility in salt solutions. And so there are certain types of compounds where you'll get liquid-liquid phase separation to occur. So here I'm showing the separation phase transition, and then we can go from two phases back to one phase through the mixing phase transition. And so there have been a lot of studies done looking at different characteristics of liquid-liquid phase separation. So on the left-hand side, on the bottom, I'm showing two different review articles, and then some seminal work on the right-hand side, in addition to the reviews. And so basically, if you take salts, water, and organic compounds, you can characterize what organic compounds will undergo phase separation by how oxidized they are or how soluble they are in the salt solution. And when they undergo phase separation, they can do it through two mechanisms: nucleation and growth, which requires an activation barrier to be overcome, and spinodal decomposition, which doesn't. And I will talk about both of these phase transitions in more detail later in the talk. And then your final structure can either be a core shell structure, where you have an inorganic aqueous phase inside and an organic rich phase on the outside, or your organic phase can form a lens on the inorganic rich phase. And we call that a partially engulfed structure. So I used to say recently, but this was 5 years ago, it was shown that you don't necessarily need a salt to have phase separation occur, that if you have a mix of organic compounds of different solubilities, you can, as you raise the relative humidity, so you introduce more and more water into the particle, you can get demixing to occur between the different organic compounds as well. Okay. So what I was interested in was, like, all the work that I've shown you so far was done on particles that were tens to hundreds of microns in diameter, which sometimes I'm going to refer to as a bulk system. And so I really wanted to know what happened to phase separation in the submicron regime. And the reason that I was interested in this is because we know that material properties change in confinement. So on the left-hand side, I'm showing two examples from art history. So the top is showing a funeral mask of Tutankhamun, and you can tell just visually by looking at it that it's made out of gold. But the bottom picture is of medieval stained glass. The red in the stained glass is red because of gold nanoparticles that are present. And so just by changing the size of the gold, you're getting very, very different optical properties. And one of the most well-known examples in chemistry of material properties changing in confinement is the example of quantum dots. So for quantum dots, we change their size very finely and we get very different fluorescent properties. It had also been shown in aerosol particles that you can get phase transitions to shift in confined volumes. So this is work showing theory and experiment looking at efflorescence and deliquescence again. So you can see the difference. So this is looking at the hysteresis loop between efflorescence and deliquescence, between crystallization and water uptake. And you can see that when you have the 6 nanometer particle, the transitions occur at a higher relative humidity than if you have a 60 nanometer particle. And what's occurring is because vapor pressures are different over smaller particles and over larger particles, you get a difference in solute concentration, and that leads to this shift in the phase transition. Okay, so what we do in our experiments is, or what we did initially, I will revise this as we go, is we take an aqueous solution of the components that we're interested in studying, we atomize it, which breaks it into droplets. We dry it through any number of different methods. Here I'm showing you a diffusion dryer, and then we impact it on TM grids. The particles are made in ambient conditions, they're made in the gas phase, and then we're imaging them cold. So we're imaging them using cryo-TM so we don't damage the organic compounds. And they're dried out completely, but we're inferring back based on their structure of what happened in the aqueous phase. And that is something that I also revise as we go through the talk. So what we saw was really surprising. We saw a size dependence of the aerosol morphology. So this is showing for just a system of ammonium sulfate and pymalic acid. So a very simple system of a salt and a dicarboxylic acid, a phase separated particle. And so the white ring is because we underfocus to get better contrast. But you can see that there are 2 regions of gray separated by a phase boundary, and that this is forming a partially engulfed structure. And then when we're sufficiently small, we just see a homogeneous particle. And almost all systems that we look at are phase separated at large sizes, and then at sufficiently small sizes, they're homogeneous. And you can see this more distinctly using a histogram. So here I'm plotting the number of particles versus their area equivalent diameter. And you can see that the small particles, the red ones, are all homogeneous. So under 30 nanometers or so, they're homogeneous. And all the larger particles are phase separated. And then there's a small region where we observe both morphologies. And because of the size regime of underneath 30 nanometers, we think that this difference in morphologies is important for the growth of new particles in the atmosphere, as well as the formation of cloud condensation nuclei. So when we saw that result, we were really interested in what was going on in terms of physical chemistry. So one of the questions that we want to ask is what actually happens as a function of drying rate? So how much of this process is kinetic versus thermodynamic? And we had started, I showed a result from a slow drying rate, but we had started at very fast drying rates. And the way that we looked at what was going on was we looked at where the largest homogeneous particle was and where the smallest phase separated particle was to get a sense of the width of diameters over which we saw both morphologies. And so when we worked at fast drying rates, we see the values that are over on the right-hand side of this plot. So this is diameter versus drying rate. And then as we dry slower, then we see that those two values are coming together. So our region where both morphologies is found is getting smaller and smaller. But because of the shape of this curve, it looks like even if we dried infinitely slowly, we would still have a size dependence of the morphology. So this points at there being, kinetic effects if we're drying fast. I will describe this in terms of activation energy. So there is a kinetic component of this, but because it would happen anyway if we were going infinitely slowly, there's a thermodynamic component of what's going on as well. Okay. So I told you that I would talk to you more about mechanisms of phase separation occurring. So this is a cartoon of a phase diagram showing water activity, which is equivalent to the relative humidity if the particle is in equilibrium with its environment, versus the inorganic fraction, the dry mixture. So how much salt we have compared to how much organic. Where you see that it's white, clear, that means that one phase is stable compared to two phases. So we should only have one phase. Where it's blue, two phases is the most stable state of the system. But we could end up crossing over the binodal, and then the system will undergo phase separation as long as the activation barrier is low enough for it to do so. Or we can go directly into the unstable region if we go through the critical point, in which case there's nucleation, in which case phase separation proceeds by spinodal decomposition and there's no activation barrier. So we ran an experiment where we're looking at what happened if we went through the binodal versus going through the critical point. And we saw that the exception that we have to most systems have the size dependence of morphology is if we go through the critical point. So this is a system where we're going through the critical point. We observe no size dependence. All the systems undergo phase separation at all sizes. And so what this means is that the reason that we observe a size dependence is because small particles can't overcome the activation barrier to form a new phase. So there's a penalty related to forming a new interface that the small particles cannot overcome. Okay, so with all that physical chemistry figured out about why this process is occurring, we looked at some atmospheric applications, and this is the one that I'll show you. So this is looking at how cloud droplets activate, how aerosol activates into a cloud droplet. And we're able to work with compound particles that were exactly the same composition, but different morphologies. So some were homogeneous, some were phase separated. And so this is looking at the fraction that are activated into cloud droplets versus the diameter. And so what we're seeing, if you just look at the green line versus the orange line is for a phase separated particle. So it's acting more hygroscopic than the homogeneous system. And that's because for the phase separated system, the ammonium sulfate has access to the air. This is a partially engulfed structure, so it activates just like the ammonium sulfate. So it acts much more hygroscopic than the particle that's well mixed. Okay, so now I'm ready to show you our recent data. I'm going to keep an eye on the time. I want to make sure that I get to the last of the 3 studies, but the first 2 should go more quickly, and the last one's a little bit slower. So these are, what I'm presenting is the thesis work of Teresa Kuczynski. One of the questions that we had based on the work that I've shown you is that everything that I've shown you before has been one organic compound, one salt, and water. One salt is not unreasonable in an atmospheric system, but one organic compound in an aerosol particle is very unlikely. We really need to consider what happens with having hundreds or thousands of organic compounds. And so that's what Teresa set out to do first. So what she did is she took a variety of different systems and looked to see whether we observed a size dependence for them. So the top 2 images here, I'm showing you ammonium sulfate and a mixture of 10 organic compounds with different functionalities that are meant to mimic atmospheric aerosol particles. At small sizes, she sees homogeneous particles. At larger sizes, she sees phase separated particles, as you can see from these images. We're changing TMs as we go, so that's the image quality is much better on these than the ones that you've shown before. We've moved from more basic TMs to fancier TMs as we've gone. And then the bottom picture is showing an oxidized terpene, so alpha pinene, which comes off of pine trees. And we expose it to ozone and just let the reaction go. And then we extract the water soluble components of that. So this is a mass of different organic compounds mixed with ammonium sulfate. And you can see, again, small particles are homogeneous, large particles are phase separated. And so I'm showing on the left-hand side the range of systems that she worked with. And the two that I just showed images of are highlighted. So you can see for both of them, I'm showing you the histograms again of the number of particles versus their area equivalent diameter, showing you small ones, the red ones are homogeneous, the blue ones are phase separated. These are fast drying rates, so there's a bit more overlap in where we see both morphologies. And so we are actually observing a size dependence for complex mixtures. We also observed some interesting behavior. So one is that there were some systems that we worked with that were 3 phases. With the compounds, particular compounds that we were using, they all had good solubility in water. So we believe that this was likely a kinetic effect. We also saw images of this weird channel morphology. And Teresa was able to confirm that that's happening at the crystallization step. So the image C is showing a picture where she flash froze a particle at a given relative humidity, showing that it's just a nice core shell particle, whereas the other two show this channel morphology, but they've been through efflorescence. So sometimes we see this in TM images of ambient particles, and now we know that it's an effect resulting upon efflorescence. So basically all systems that she looked at had a size-dependent morphology, which is suggesting that the size dependence will also be present in ambient aerosol. And there are 2 unique morphologies that we observed, one with 3 phases and one with a channel morphology. So Teresa also set out to accomplish something that we, another problem that we're having with the previous systems that we'd worked with, which is that everything that I've shown you so far, we had to dry in order to be able to infer what was happening in the aqueous state. And we really wanted to capture particles wet to understand what was actually happening in the aqueous phase itself directly. And so she developed a flash freeze flow tube for the study of the phase transition process. And the way she did this was to have a Tedlar bag. Tedlar is similar to Teflon, where she can let the particles equilibrate at given relative humidity. And then a flash freeze tube that can freeze the particles over the course of several microseconds. And then we capture the particles cold and we image them cold. So we calculated the temperature profile. So and that allowed us to calculate how quickly things were freezing within this chamber. And then we were looking at the efflorescence of potassium salts. And we looked at potassium salts because they had been previously studied in the submicron regime. And so we were able to know what their efflorescence value should be. And so you can see on the left-hand side, images of aqueous particles. So these have lower contrast. And in the top image, it looks like an aqueous particle has hit a grin, which is exactly what happened. The bottom image also looks aqueous. And as you decrease the relative humidity, you can see that they look more crystalline. And so we're able to measure where efflorescence occurs. We can also look at liquid-liquid phase separation. So this is looking at 2-methylglutaric acid and ammonium sulfate. We would expect them to undergo phase separation at 75% relative humidity, but lo and behold, at 71% relative humidity, she has an aqueous particle. When you continue to lower the relative humidity, you do eventually see liquid-liquid phase separation occur. Here you see a core shell particle. As you decrease the relative humidity more, you can see that you're getting a partially engulfed particle. And so this transition between core shell and partially engulfed has been seen in the literature previously. We know that what morphology we have is really sensitive to the water content of the particles. Okay, so this flash freeze flow tube allows us to track the separation relative humidity for submicron aerosols I showed in the last example. And so we can determine if different results are seen compared with particles that are tens to hundreds of microns in diameter. And so that was the last study. Looking at the dynamics of the phase separation process using this technique. So when I was an undergrad, I did a directed reading during my junior year using Chemical Kinetics and Dynamics by Steinfeld, Francisco, and Haase. And that really inspired me to study dynamics as a graduate student. And ever since then, I've been trying to get back to dynamics, although our work hasn't been in that area until this paper. So this is a really exciting paper for me because it gets us back to looking at dynamics, although it's not really cross-beam molecular beam dynamics like I would have read about in that book, but it's still looking at the process of how things are actually occurring, which is exciting to me. So here's our phase diagram again. So what we're doing in this experiment is we are going through the phase diagram again. We're starting at high water content, and then we go vertically through the phase diagram at a given composition of particle. We know that if we start high enough, we should only see homogeneous particles. We know that if we're low enough in water activity and in relative humidity, we should see a size dependence because we've been able to look at those extremes before, but we don't know what happens in between. So this is looking at a mixture, that same mixture of 10 organic compounds mixed with ammonium sulfate. And I'm showing the images similar to what I showed you before on the left-hand side. So at 83% relative humidity, everything is homogeneous. At 66%, you can see that you start to get inclusion. So phase separation has happened, but everything hasn't coalesced together. At 51%, you can see that you have a nice core shell structure with one core. And then at even lower relative humidities, it's shifted from core shell to partially engulfed. On the right-hand side, I'm showing the summary of this data. So this is showing the percent relative humidity versus diameter. So what happens in this experiment is that we're equilibrating at a given relative humidity and then capturing those particles. So every relative humidity that we do is a different experiment. So these are rather hard experiments to run. And that's why you see just these individual lines running across. The black line is showing you where we would expect for much larger particles like droplets. So the bulk value of the separation relative humidity. And you can see when we image particles right under that value, they're all homogeneous. As we go lower in relative humidity, you can see that we observe phase separation. So that's indicated by the blue diamonds. And if you look at all three of these, it looks like there might be a size dependence for these top two, but the bottom one looks random. We never have the smallest particles undergo phase separation. And when the phase separation is like mature, then we see the size dependence. So here, all the small particles are homogeneous. All the large ones are phase separated. There's a little bit of overlap. So just to summarize this picture, there's a lot of data here. It shows that phase separation takes place at a lower relative humidity than measured for droplets, tens to hundreds of microns in diameter. And it also shows that the phase separation process is occurring over a huge range, like 10% relative humidity, rather than being a prompt transition. And so we did this for 3 different samples, ammonium sulfate mixed with this mixture of 10 organic compounds, which I just showed you. And then the middle image is showing 2-methylglutaric acid. The one on the right-hand side is showing 1,2,6-hexane trial. The black line is always showing what the bulk value of the separation relative humidity is. And methylglutaric acid it starts to undergo phase separation at a much lower value than that. 1,2,6-hexane trial is a bit closer. It's still under that value, but it's closer. And we need to investigate further why that's occurred. And you can see from the middle and the right-hand side like images, you can see that the phase separation really is random with size. There's not really a size dependence to what's going on. And then eventually at the bottom, you see the size dependent morphology that I've shown before. So again, phase separation takes place at lower relative humidities than measured for droplets and over a larger range of relative humidities. So our hypothesis based on these results was that we were observing what we were observing because this was an activated process. So when we have nucleation occur, molecules diffusing through a solution need to form a critical nucleus if they're going to phase separate by nucleation and growth. And so if the process depends on forming that critical nucleus, if we have more time to go through the phase transition, more particles should phase separate. That's our first hypothesis. And our second hypothesis is, again, if the process depends on the activation barrier, then if we remove that activation barrier by going through the critical point, the phase transition should proceed promptly. And so this is looking at equilibration time. So what we did was we had the particles equilibrated at about 5 minutes, where you see that there are more homogeneous particles and phase separated particles. Then at 20 minutes, where you see that they're about half and half. And then at 75 minutes, where many more are phase separated than are homogeneous. So lo and behold, if we wait longer, more of the particles phase separate. So this indicates that phase separation process is proceeding through the formation of a critical nucleus. And then we're able to also go through that critical point and have the phase transition occur through spindle decomposition. And when that occurs, so here we have all the homogeneous particles, and then basically all of them phase separate. A few of the large ones stay homogeneous, but this is a pretty prompt phase transition compared to what I've shown you previously. And we also see that where we would expect, based on the bulk separation relative humidity, that equals what's happening for these submicron particles. And this also supports the behavior seen on previous slides is due to this activated process. So, so our results show that phase separation in submicron particles is differing from bulk systems. And in that we're seeing phase separation at lower relative humidities and over a larger range than for larger droplets. And so that could be indicative of an effect of small sizes of particles, or might indicate a difference in activation barrier, suggesting a shift in the binodal curve. So that's what I'm showing here, that when we have that the critical point is occurring at the same relative humidity, although based on previous work, we know that it might be slightly different in composition, but that our binodal might have shifted. And what I'm indicating by the regions that are shaded is that the bulk separation relative humidity is occurring over a very small range, whereas for submicron particles, it's occurring over a much larger range. This suggests both, because we're getting different values of separation relative humidity, it suggests that we need to go back for modeling to get new parameterizations of what's going on. But it also suggests that phase diagrams for these submicron particles might be different than what we've seen for larger particles. And so to conclude this section, I think I've said the first 2 points like 3 or 4 times, but basically we're observing behavior that's a result from nucleation occurring through an activated process. But it's exciting to observe that directly rather than being able to infer what happened in the aqueous state by what we're seeing from the dry particles. And so in conclusion, so liquid phase separation does not occur in sufficiently small particles, because small particles cannot overcome the activation barrier needed to form a new phase. That's what I showed in the background of the talk. And then the 3 projects that I showed show that these results of the size dependence morphology apply not just to simple model systems, but also to systems much closer to atmospheric models. I've shown the development of a flash freeze chamber allows us to capture particles equilibrated at a given relative humidity, vitrify them, and then capture them for imaging. And that this then allows us to look at the separation relative humidity, which occurs at lower relative humidities and over a larger range of relative humidities than observed for bulk systems. And with that, I'll conclude the talk, and I'm happy to answer any questions that you have.
Very nice, Miriam. That was outstanding. Perfect. Okay, so it looks like we have a fresh question from someone in the audience, but maybe Carlos and Rodrigo, which one of you would like to go first, and then I'll fill in the question that's in the chat there.
I can get started.
Okay, great.
Thank you for such a great talk, Professor Friedman. I have a couple of questions. I'll ask one, and then if there's time, I'll ask my other one. So you mentioned that cloud nucleation depended on whether the particles were homogeneous or if they were engulfed in the morphology. Is this a binary either or type situation, or about being exposed as like the ammonium sulfate phase, or is it something that depends on the shape and the nature of that three-phase interface between the two phases in the particle and the vapor?
So when you do that type of experiment, what you're doing is you're taking the particles and you're exposing them very rapidly to a supersaturation of water vapor. So any water vapor in the air is just going to clump onto the particle very, very quickly. So if there are effects of shape or like small heterogeneities, I don't think you're going to see them. It's just such a rapid process.
Thank you.
Okay, I guess I'll ask my question first. Thank you again for the wonderful talk. I really enjoyed this, and it's a new area for me. I don't know very much about atmospheric chemistry. So my question is related to maybe it might be a little bit outside of your talk, but I remember some recent results from Dick Zer's lab at Stanford showing that there are very large electric fields in these micro droplets, and those electric fields can actually completely change the kinetics of reactivity within the micro droplets. So my question is, presumably the similar fields are present in these droplets, and do you think that that could affect the thermodynamics as well as the kinetics of the liquid-liquid phase separation?
So the work that I'm aware of that shows, so some of the work that shows acceleration of reactions in droplets has been done using electrospray, where you're purposely putting a lot of charge on a droplet. I don't think that there's that much charge on an atmospheric droplet, although for it to have some charge would not be unlikely. That's really an understudied area within our field. So I think if you're talking about a little bit of charge, to see that effect would be very interesting, but it's unlikely to have a lot of charge. People are really interested in the atmospheric community at acceleration of reactions in droplets and aerosols. That's I would say like not something that's been studied to a great extent, but something that's starting to be studied. And so that is of interest.
Thank you.
This question's from Paul Ono. Paul asks, first he says, thanks for the really nice talk, which is correct. Is there any difference in interfacial tension between the glutaric acid AS and the hexane triol AS? And could that play a role in why one deviates more from the bulk behavior than the other?
All right, so that's a great question. So one of the and we have measured surface tensions of the systems that we look at. The issue that we have is that when we look at surface tensions, we tend to be looking at the equivalent of like a 90% relative humidity solution, because what happens as we're undergoing phase separations, often phase separation occurs in a metastable state where we're super saturated with respect to components. And so we can't measure the surface tension easily. Like some people measure it using AFM, but we can't measure it using a bulk system at the conditions required for like where phase separation is actually occurring. And so when we're measuring them, like when we're measuring surface tensions at 90% relative humidity, the water washes out most effects. You just have too much water in the system. So what's happening at the point of phase separation, we don't know. And even if we tried to go after it with an AFM, there's a chance that we would like nucleate the new phase when we weren't intending to, because we've introduced like heterogeneous into the system. So it's just a really hard measurement question, a good one, but yeah, where we can measure those effects are wiped out if they exist.
So maybe we'll take one of my questions, which I guess I'm going to ask another, things for the future. Your fancy flash freeze instrument allows you to access now just a whole new range of variable conditions. And in addition, right before that, you talked about the really complicated organic mixtures where you had like 10 different constituents in there. So the other thing, though, that's present there is all the inorganic, components of the atmosphere. Is anybody else, looking at more complicated mixtures, including the inorganic, both compounds and the ions, or is that something you have planned to do as well?
So we have looked at the effect of different ions on the phase separation process. So I should say Alan Bertram's group has looked at it for supermicron particles. We've just recently looked at it and we'll be submitting a paper imminently, probably on Monday, on submicron systems. And I'm actually going to present those results as part of my talk at ACS in the fall.
Awesome. And hopefully you reference a lot of JPC and JPCL papers. So hopefully those manuscripts that when you're submitting come into Journal of Physical Chemistry letters. Let's see. Rodrigo, do you have another question? It looks like there's one more in there.
I was just wondering how this might apply more to the earlier results that you showed as opposed to the flash freeze chamber ones, but how you would draw a connection between those single particle studies and situations where the particles are able to interact in some way and coalesce, for example, and things like that.
So I think I need to know, like, in terms of what. So if you have droplets that are coalescing and they're still in the aqueous state, they can just coalesce together. Jonathan Reed has had some interesting studies about what happens when you do that when they're like at different viscosities, and measuring viscosity through looking at coalescence processes. But if they're liquid-like enough, then they'll just coalesce and you'll get phase separation of the whole system. And then if they're drier, they can aggregate. But I need a little bit more context to answer what I think you might be asking.
Yeah, it was more about the way in which the demixing process might be affected if there's another thing driving the kinetics.
Okay, so like introducing something that might alter the phase transition, the ability to undergo the phase transition. That's an interesting question. So we're starting to look at like different properties of systems where where things are mixed together versus separate. And I have a colleague who has the ability to like mix them over a long time scale. But we haven't really looked at whether we can drive or the demixing through coalescence processes, but that's interesting.
Thank you.
Great. So for the the bigger part of this conversation, I feel like we are exactly on time. And so we should thank Professor Freedman for really an outstanding talk. You did just a great job of framing that in terms of things that other physical chemists who aren't exactly in your field can understand, but then also demonstrating how you're leading in the field. And so that's an example for all of us to try to do a good job of that when we give a talk. So thank you.
Thank you.
And then at this point, if all of the faculty attendees would choose, yep, I see people checking out. And if anybody who's a student or